Welcome back to the course Fundamentals of General Immunology. Today we will try to learn few of the proteins or molecules which are important for the immune regulation, and those were not discussed in the lectures. So we'll try to understand those proteins and how they are relevant in the context of immunology. That is what we'll see in this tutorial. That is what we learn in this tutorial. So when we see that there is an infection in a cell, host cell, so say for example if this is a cell and if there is any infection, and that infection could be a virus or a bacteria or a fungi. Now in any of these cases the host response will be generated to fight against these molecules. So the host will produce a lot of molecules which can block the replication of virus, bacteria, fungi in order to protect the host. Now since the host is not exposed to those pathogens earlier, these all molecules are non-specific. So this non-specificity is what is unique about these molecules which are regulated upon the exposure of this virus, bacteria, fungi or a pathogen. So what it implies that the host is having all the necessary information in their genome. Say this is nucleus, so all the details are there in the nucleus that allow the production of these non-specific molecules upon the entry of this pathogen in order to protect the host. While once the host is sensitized with the virus, for example now virus and bacteria or fungi, anything is already infected the host cell, infected host, then probably what we will see is there could be a generation of a very specific antibody. Now these antibodies are very precisely generated against a specific antigen. They can bind to a very specific antigen. So antigen could be very specific which can bound to this particular antibodies. Now these response we can call it specific because these are generated post-infection. So these are all post-infection response. Now we are trying to understand one basic fundamentals of a host system where the immune response is divided into two parts. One is having non-specific and the one is specific. When it comes to non-specific, that is what is coming under the innate immune response. So these are all innate, and this is what we call adaptive, which is coming later in the life post exposure to the pathogen. So we have understood multiple pathways, multiple proteins which are responsible for the host immune response. Let's try to understand one of the very specific molecules or the protein which is required in order to fight against the pathogen and generated by the host body most oftenly upon the virus infection, and that's called interferons. So we'll try to learn about this interferons in today's tutorial, how these interferons are produced, what are the relevance of the interferons, and how they can get rid of viral infection by modulating the host system. So we'll see this interferon in details in today's lecture. So this protein was first identified in 1957 by two scientists. One was Isaac and Lindenman. They identified this particular protein when they were doing an experiment with influenza virus. What they observed that once you take a chorioallantoic membrane. Now if we look into the egg of a chicken, so suppose if there is a egg, then we see that there could be an egg shell. So this is an air sac cavity, and then we can have a membrane that's lining the whole egg covering. So this layering is called chorioallantoic membrane, CAM in short. And then we see that the embryo can grow internally here, and then we can also see there could be a yolk sac, and then also the blood vessels. So basically we can see the development of blood vessel in embryo by candling these eggs. So we generally use this membrane and the fluid which is present here. This is called allantoic fluid, because here it is a chorioallantic membrane. So this is called as allantoic fluid, and this area is used to grow multiple viruses and see that what are the changes that we can see in the embryo post virus infection. So this is very unique bioincubator, a sterile bioincubator, one can say. We can use this embryonic egg to see multiple things. So one of the experiment which people started doing is taking this layer of chorioallantic membrane and see how we are infecting the virus and all. So what Isaac and Lindman did is they took this CAM, chorioallantic membrane, and then infected with a heat inactivated influenza virus. That means the influenza virus which may not be able to cause disease condition. So once you have a virus, there are ways to inactivate the virus. Either we can give a heat treatment, we can give a chemical treatment as well, or we can use UV irradiation. Now all these three processes will eventually make this virus little attenuated. So virus won't be able to cause the infection. This is what you mean by treatment and inactivation. So this is a live virus which can be treated by heat, UV or chemical, and create the different variant which may not be able to sufficiently infect the host cell. That is what these two scientists did. So they inactivated the influenza virus and then infected into the chorioallantic membrane. So and then they incubated the virus and the CAM together overnight. So what happened that since virus is unable to infect the membrane, but the cells which are present in the membrane, now remember these are all cell mass actually, so membrane is made up of multiple cells actually. So the chorioallantic membrane is sensitized with the heat inactivated influenza virus, and then they generated few things actually upon this infection which was there in the media, and that is what is listed here or shown here. So these materials actually that were not identified at that time. So what they did is they discarded the chorioallantic membrane and then took those supernatant which were containing all those materials which were generated by the chorioallantic membrane after heat inactivated influenza virus infection, or I would say treatment, because there is no infection. Now once they took those materials, the supernatant material, and then they freshly incubated it into a fresh membrane, fresh chorioallantic membrane, then and again incubated overnight, and then on the next morning they infected with the live virus. So we expect that the virus will cause an infection, but just look into the prior exposure. They were priorly exposed to something which came from the cells which were infected with heat inactivated virus, and those all material gave something to this cell which allows the protection to the cell population. That means the chorioallantic membrane, even though they are infected with the live virus, won't get any infection because something happened in the cellular system. So they did a lot of experiment to prove that these are the protein which can interfere to the growth of the virus into a new cell type, and therefore they call those all interfering molecule as interferons, so which can interfere the virus infectivity. So this was a discovery, an outstanding work where people looked into carefully that post infection upon inactivated virus, the cells can non-specifically, which we discussed in the prior slide, that non-specifically created some protein into the cellular system in order to protect the cell. Since the virus is unable to infect, there is no pathogenicity or no as such infection, but these proteins were generated. If these proteins were priorly sensitized to the host cell, then we won't see any infection to happen in the cell. and that is what is the beauty about this system. So this is non-specific. Remember, when we call it non-specific, doesn't mean that it can only protect against influenza virus, because this is a non-specific protein. So the protein can act against any virus or any pathogen. That is the meaning of non-specific. So remember, the interferons are non-specific. They don't differentiate between a virus. They don't say that, well, I can only neutralize influenza virus because my generation was from the influenza virus. No, because it can neutralize any other virus as long as its ability to neutralize that molecule. So it's non-specific. Because when we discuss immunology, we always discuss about the specificity, how specific antibodies are against a particular antigen, but interferons are not. They are non-specific. Now, let's see how these interferons can cause an antiviral stage, and of course we'll go in detail and see the molecular basis of those inhibition as well. So once the virus can infect the cell, we see that the post-infection, the virus will generate these interferon proteins, and that protein is secreted by a particular cell to the environment. What is the function of that? Now this cell is already infected, so probably this cell is gonna die, but the information that's coming out from the cell can pass on to the neighboring cell, so that the neighboring cell can be sensitized that, look, there is a virus infection happening in the nearby, and better you can protect yourself. And therefore this is kind of an altruistic behavior by a particular cell that's dying because of a virus infection. So interferons can be segregated or secreted in the nearby tissue, and if they go and bind to the receptor which are present on a cell, so suppose this is a cell which expresses the interferon receptor, the interferon molecule can go and bind to those cells, and then these signals can pass on to the nucleus that these interferons are actually a stress molecule. They can't reproduce all the time because there is a virus infection. Gene those are against the viral infection will be switched on, and therefore the signaling can allow the switch on phenomena where the gene transcription become active, and once there is a gene transcription, we see that the proteins are coming out. A lot of things will come out along with the gene transcription, and this protein can block the virus replication by multiple ways. Either they can block the entry area, so they won't allow the virus to bind to the surface. There could be a breakdown of the viral messenger RNA. Say there could be messenger RNA which can be generated post-infection. Again, that can be broken down into smaller pieces so that the protein won't form in a proper way. So the first thing is they can block the entry of the virus. If they are not successful, even though virus is going inside some of the viruses, because viruses are more in number, once they are infecting, they create a lot of virus progeny. So when the virus is entering, they can immediately produce the protein, and before protein they can form the messenger RNA. So the messenger RNA can be broken down so that the protein won't form. Even if there is a protein synthesis, they can stop the protein synthesis by halting some of the ribosomal protein as well. So even if there is a transcription, so there is a steps actually. Suppose the blocking is not possible, then there is a transcription stage. If the transcription is bypassed, then you have a translation stage. Even if the translation is passed, then you can block at the level of their release. So each and every stage, interferon can cause inhibition of the viral particle. Either they can block the entry of the virus particle, transcription of the viral protein, translation of the viral protein, or budding out of the viral protein, the matured virion. If they are unable to go out, they won't do an infection. So they can be restricted, and that's the beauty about this system, which is non-specific in nature. So what we are trying to understand is about a protein called interferons. These are produced non-specifically upon a virus infection. Once they are produced, the cells which are producing those interferons are actually going to die because they are already infected with the virus. But the interferons which are secreted into the atmosphere or the nearby environment can go and sensitize the neighboring cells by binding to the receptor, and then these receptor binding of the interferon can start the signaling cascade which can turn on the gene that are responsible for producing lot of proteins, which we'll see in a while. Those are all called as interferon-stimulated genes, ISGs, because the genes which are turned on is because they were sensitized by the interferon. So therefore, these are called interferon-stimulated genes. And there are many numbers of those interferon-stimulated genes, and each function of those interferon-stimulated genes are very well explored in a research point of view. So, and then finally we see lot of inhibitory pathways can be activated in the cell post these genes turned on, and then we can either see that they can block the virus binding, they can block the transcription, translation, or budding. Eventually they can lead to the inhibition of infection towards the particular cell type, and that is how the cell can be protected against a virus infection. So these are non-specific. Now, since we are talking about this biology, we need to understand the signaling cascade. These are very important. How the signal can pass from binding of the interferon to the downstream nucleus so that they can start transcribing the interferon-stimulated genes. So let's see that part also. So interferons are of different types, and these are depending upon their action, how they are produced, how they can stimulate the cells, and what are their effector function altogether. So there are three types of interferon: type 1, type 2, and type 3. And these types of interferons are having multiple different types again. So interferon gamma is actually interferon type 2, while interferon alpha and beta is basically major interferons are only 3, alpha, beta, and gamma. So alpha and beta is type 1 and gamma is type 2, while there are other interferons which are also coming under the class of type 1. And we also have a separate class of interferon, class interferon 3, type 3 interferon, which is interferon delta. So there is a separate class of interferon, and that's called interferon type 3, and we have a separate interferon lambda for that class. Now we know that there are multiple interferons which are there, and their pathways to stimulate the gene is also different. So let's see one by one. So let's start with the type 1 interferon. So these are type 1 interferon specific receptor. So these are receptor, and once the receptor can bind to the interferon, we see that the downstream cytoplasmic tail of the protein is having some kinases. These kinases could be Janus kinase or tyrosine specific kinases. So there are kinases which can do phosphorylation. The function of the kinase, so the kinases are actually a protein. So kinases are protein. They can add a phosphate group by using ATP. So ATP can be converted into ADP, and the phosphate group can be tagged into a protein. So basically you have this protein which will be bound to this phosphate group, and then we get a phosphorylated protein because the ATP can donate a phosphate group to this protein, and post phosphorylation we see this protein become active. Before that it is non-active. So therefore kinases are very important protein which can allow the addition of phosphorus group, phosphate activity, to make the protein phosphorylated. So Janus kinase and the tyrosine kinase are two such kinases which can do phosphorylation of the protein that can start the transcription process, and these two proteins are STAT, signal transducer and activator of transcription. We have looked into this protein multiple times, so we will not discuss about those. So these are specific transcription factor. Once they get phosphorylated, they can form a dimer, because once the STAT1 and STAT2 become phosphorylated, they can form a dimer, and then in addition with the interferon regulatory factor 9, which is listed here, they can bond and make a trimeric structure and then go inside the nucleus. So this is actually a nucleus, and once they entered into the nucleus by this nuclear pore complex, so this is nuclear pore complex, once they go, they will bind very specifically to these ISRE, which is interferon stimulated genes specific or interferon stimulated response element. So they can specifically go and bound to those genes which can So these are promoters of those genes. So this can be a gene, and these are actually a promoter of that particular gene. So they can bind to the promoter so that to start the transcription of that interferon stimulating genes. These trimeric structure is called as interferon stimulated gene factor 3, ISGF3, which can start, well, name is not important to learn or remember, but just to see that how these cascades are activating. So what we are trying to see that how we can activate this interferon stimulating genes post binding of interferon to the receptor. So once the protein can bind to receptor, it can activate kinase. These kinases can do phosphorylation of the protein, which can form a dimer, and then combined to the IRF9, they can migrate themselves to the nucleus in order to bind to the promoter, which can allow the transcription of a gene. Very complex biology. But anyway, that's what the function of interferon is. So once they can activate this interferon stimulating gene, they can do multiple function. Similarly, in type 3 and type 2 also the similar biology can happen, while type 2 is mostly the homodimer. So you see that STAT1 is actually a homodimeric form, while here it is a heterodimer. So you have a STAT1 and 2. So remember, type 1 interferons are mostly activated by heterodimeric form of the STAT protein, while type 2 is mostly the homodimeric form because both are same. Function is almost same because the Janus kinase can do a phosphorylation, they can migrate, and they can go and bind to this gamma activated sequence, which is again a promoter, but this can regulate a different set of interferon stimulating gene, while they can activate a different set of interferon stimulating gene. The function is same because once they are coming out into the system, the whole function is to make the cell immune against the entry of viruses. So essentially these are very, very important regulatory control a host cell can do post virus infection, and if this fails, then the person starts suffering from a disease condition. So this is non-specific internal control of the host system. So this is what is the signaling biology of interferon. So remember, once in the broad way, once the protein is bound to a surface, it can do phosphorylation, and once the phosphorylated protein can migrate to nucleus, it can start the transcription of genes, and these genes can again come out, the messenger RNA will come out into the cytoplasm, bound to the ribosome, and then produce a protein, and this protein could be coming out to make an antiviral function. And these all proteins are actually interferon stimulated proteins, and these are coming from the interferon stimulated genes. All right, we'll move on and we'll see the basic biology of those proteins. So we discussed that the protein can do antiviral activity. How they can do an antiviral activity, that is what we need to understand. So once we see that the proteins which are coming out, so we'll just draw a diagram here. So what is happened that interferon can bound to a receptor, and they can secrete this protein which is coming out after the transcription of these ISGs. So this is ISG specific protein. So we are trying to see the biology of this protein that can do an antiviral effect. So once these proteins are coming out, they do multiple function. There are many proteins which are well documented that can be antiviral, and people are trying to explore this biology in order to treat patient with a virus infection. Many proteins were studied during the COVID time, where people started looking into, can we boost the immune response of the host cell to fight against the COVID infection. So one such protein is PKR, protein kinase R. So protein kinase R is actually the kinase, so they can be phosphorylated, so they can itself be activated by multiple biology. So once there is a double-stranded RNA can activate the PKR pathway. Once the PKR is activated upon interferon, this PKR is actually known to phosphorylate the eukaryotic initiation factor. So once there is a protein synthesis, you require to have some. So suppose this is a ribosome and this is a messenger RNA. So essentially we will get a protein, and how we get a protein is the transfer RNA can read the codons which are present by its anticodon loop. So this is transfer RNA, and this is messenger RNA, and the messenger RNA is from the 5' to 3' direction. And this is, say for example, this transfer RNA is having this anticodon loop, and they can bind to this, the codon in the messenger RNA, and then they can put an amino acid. So accordingly this amino acid will be linked, and then we see the protein function. To start this process, we have this protein called eukaryotic initiation factor. So protein translation won't possible unless until we have all the factors available there. So one such factor is EIF2 alpha. EIF2 alpha. Now EIF2 alpha is active, but if it is getting phosphorylated, then this become inactive. So once the PKR is activated upon the interferon stimulation, this double standard virus can also activate the PKR by the biology, which is kind of somewhere explode. Then we see that the eukaryotic initiation factor is getting phosphorylated. Once there is a phosphorylation, that the protein synthesis is getting inhibited. Now what is happening? That once there is an interferon, so we can largely see that, suppose this is a cell, it is attacked by this interferon, and so once we know that these cells are attacked by this protein, which are called interferon-stimulated gene-specific proteins, they can do a global shutdown of the protein synthesis. So the protein synthesis will be shut down. Why? Because we have eukaryotic initiation factor 2 alpha getting phosphorylated. So this is what the meaning of this first one, that once there is a interferon specific effect, the PKR is getting activated. PKR do phosphorylation of EIF2 alpha and make the protein synthesis inhibited, because the phosphorylated form of the EIF2 alpha won't be able to fit into this domain or cassette, and therefore there is no protein synthesis in a proper way. The second possibility is the interferon can induce the another protein called a 2,5 oligoadenylate synthetase, which can make this 2,5 oligoadenylate. So these are molecules, so these are especially a stretch of the A which require the phosphorylation from the ATP as well. So this 2,5A activation can cause an RNase L dependent lysis of messenger RNA. So basically they can do an RNase activity, RNase L activity, so they can start doing an RNase L activity. So this RNase L activity can degrade the messenger RNA very strategically. So we have this RNase L coming into the site, and once there is a production of messenger RNA, these RNase L can destroy this RNase messenger RNA in the host cell. So messenger RNA in the host cells are destroyed by this RNase L that got activated upon this interferon-stimulated gene-specific product. So we understood that PKR is a protein, 2,5 oligoadenylate synthetase is a protein. Now these two can be called as interferon-stimulated proteins, which can be generated upon this interferon stimulation, that can lead to either protein synthesis or the RNA degradation. So that means you have a transcription control and there is a translation control as well. So ultimately we see that there could be a complete host protein shutdown and host RNA transcription shutdown. So basically we don't have a messenger RNA, functional messenger RNA, because these all messenger RNA is getting degraded, and therefore we won't see any protein production as well. So there are two ways control which can happen post production of these proteins from interferons. So these are two interferon stimulating genes specific product. Let's see what are the other proteins which are very well explored in the field. So there is a list of the interferon stimulated genes. So this is either MX1, OAS1, which stands for the same which we discussed, oligoadenylate synthetase, which can form 2,5 oligo A, RNase L, which we just discussed, that can bind and degrade the messenger RNA, protein kinase R, that can do a phosphorylation of EIF2 alpha, which is here, and there are other proteins which are called IFNs, IFIT stands for interferon-inducible protein, which can very specifically degrade some of the messenger RNA, specifically the capping and all. So we'll see in a while. Then we have ubiquitin-like protein, which is also called as ISGs, and then we have BST2, which is also called as tethrin, that won't allow the virus to bud out actually. So they can stop the budding of the virus particle from the host cell. And then we have whole bunch of transmembrane protein called IFIT-M. So interferon-induced transmembrane protein. So they can just present on the transmembrane and they can inhibit the viral entry. So we started our discussion with the ways the virus can be inhibited upon the interferon production, and these are the proteins which can do all this function. So they can either say MX1 is GTPase activity, they can produce some intermediates such as 2,5-oligoadenylate synthetase, they can produce some of the protein which can either degrade the messenger RNA or can do phosphorylation. There are other proteins which can bind to viral. So suppose IFIT1 can bind to viral RNA lacking 2' O methylation at the cap, and therefore it's basically can bind and degrade the messenger RNA and won't allow the messenger RNA to block the translation of the viral mRNA that lack the proper 5' cap methylation. So these are strategy a host system will do in order to stop the virus replication. There could be other possible stage where IFIT3 can block the translation, so inhibit the translation as well. We have ubiquitin-like protein, where we see that ubiquitination is a process where suppose if you have a cell and there is a protein, and this protein is required by the cell in order to do some function. For example, if right now we are at the 37 degrees centigrade and the cells are kept at 37 degrees centigrade. So this 37 degrees centigrade requires some protein to be produced in order to make this cell sustained at 37 degrees centigrade. Now suddenly if you shift this cell to say 5 degrees centigrade, then we don't need this 37 degrees centigrade protein. So we need to degrade this protein and make some important protein that can sustain the cell at 5 degrees centigrade. That means what is happening is we have some protein that are made at 37. Suddenly we are shifting the cells to 5 degrees centigrade, and then we need some more protein to be sustained at 5 degrees centigrade. Now that's not possible to make all the time. We have a very limited capacity of the cellular environment. So what will happen, that this protein which were generated at 37 degrees centigrade will be degraded in order to make this protein. The degradation of the protein is happening in a very specific structure in a cell called proteasome. So once this protein is done with the job, they can bound to the smaller protein called ubiquitin, and this ubiquitinated protein can go and getting degraded into smaller amino acid, which can make assembled into a neoprotein. So this is a host cell phenomena where the protein is getting recycled. The older protein is getting every day degraded and the new protein is formed based on the function of a cell, and therefore this ubiquitination is an important process. So once the protein is bound to this smaller ubiquitin protein, we called it ubiquitination of a protein, and once the protein is ubiquitinated, that can go and nicely getting degraded by this proteasomal degradation machinery that's available in the cytoplasm of a host cell. And this protein, smaller fraction of the protein, can be recycled back to make a newer protein. That is the function of ubiquitination. So the viral specific protein, once they are coming into the system, we don't need virus with specific protein. So suppose if the cells are infected with a virus, the viral proteins are made. Basically this protein will be utilized to make the virus particle. Host cell don't want to make this protein available in the cell, and therefore we need to degrade it by proteasomal degradation, and that is what is done by these specific protein called ISGs 15 that can allow the protein to be degraded by proteasomal degradation, so that we won't have a functional viral protein, and therefore there is no virus infection as such. So this is a host protection mechanism by producing certain proteins. So these are very important proteins which are generated upon the interferon stimulation, and providing a multiple ways the protection to the host cell against a virus infection. So this is something very important and interesting when we are reading it. Multiple lectures which will come in future will allow us to use the interferon in order to see that the specific biology of the host cell can be altered by just using interferon or interferon stimulating genes. So that's all for today, and we'll try to learn few more things in our coming classes. Thank you.